must say I'm glad to be standing now instead of sitting. So first I'd like to thank the organizers. It's a great honor to be invited to the symposium, and in particular I want to thank John. And you don't realize this, John, but when I was a master's student around the year 2000, I saw you give a talk at Michigan State University where you talked about this bridge between population genetics and phylogenetics. And I entered the talk being a master's student who was working on this really morphological cladistic analysis of a group of bats, and I left the talk thinking I want to do phylogeography. And so thank you so much for moving me in this direction. So I was asked to talk about phylogeography's place in population genetics. And I really struggled with this topic for a while until I thought back to something that my PhD advisor, Jack Sullivan, had said. And so those of you who know Jack know that he's full of witty comments and somewhat cynical comments. And at one point he said, you know, Bryan, if you want to be really good at phylogeography, you shouldn't read phylogeography papers. What you should do is you should figure out what human population geneticists were doing 10 years ago. And then you should do that, and you'll be, really setting the pace for phylogeography. And that's a little cynical, but I think in some ways it's true, not for lack of effort on our part, but because the data that we've been able to collect in non-model systems has really lagged behind the data that you collect in humans, for example. And this has been a constraint that the field has been dealing with. And the really good news is we're catching up. We still can't collect the same quality of data as easily as you can collect from human systems, but this gap is closing. And what I'm going to try to convince you of today is that we have a great opportunity to ask different kinds of questions that are uniquely important in non-model systems that we all investigate. And so as I go through my talk, I'm going to try to bring the kind of work that we do in non-model systems and contrast it to the types of questions that are important in model systems. And I'm going to use this system of the four-eyed frogs that I've been working on with former postdoc, Teresa Tomei, as a sort of an example of this. And it's very much a work in progress. See if I can figure this out. Okay. So phylogeography is this bridge. And to me, what this means is that it's sitting at the species boundary. It's a discipline that's really interested in processes within populations that ultimately lead to speciation. And as a master's student, when I saw John give a talk that included this metaphor, this seemed really exciting to me, and it still does, I'm happy to say. So in my lab, we work on a variety of different kinds of organisms. And the typical model that I have with my students is they really come into the lab being very interested in certain types of organismal biology, and then we work together to figure out what the interesting questions are, and we try to really think hard about how we analyze our data. And so I have people working on things like bats and salamanders and carnivorous plants, and these are all students that are either graduated recently or will soon be graduating, and they need to fledge and move on to postdocs if any of you are interested. But they've been really great about thinking hard about how we're doing our analysis and why we're doing things in a certain way. And so I want to start off by asking a question that we've all asked ourselves, and that's how we analyze the data that we collect in phylogeography. I think that the field uses different types of approaches depending on the data we collect and maybe when studies are designed or what our goals are. But a lot of studies have analyzed data by making some summaries or calculations from the data that are relatively assumption-free. And so whether this is a gene tree that's interpreted in geographic space or a structure plot, they're analyses that you can make from the data without a long list of qualifying assumptions. And then the researchers will make inferences about the history of their organism from these analyses. As the field has developed, we've started to use more and more complex models that have a greater number of assumptions along with them, and the hope is that they allow us to have more power to make estimations. And so I'm going to pick on Craig too a little bit here as an example. So this is one of my favorite papers by Dolman and Moritz in 2006, and they were, not surprisingly, looking at skinks, and they were interested in understanding population parameters such as the size of the population, the timing of population divergence, and the rates of gene flow. And they wanted to do this so that they could understand how the skinks had persisted throughout these glacial episodes. Even though they had this very complex model, and at the time this was like, I don't know, 6 or 8 loci, it was one of the leading studies with sort of multi-locus data and a model that could actually analyze these data. They made these estimates from the data, and I've highlighted here the migration estimates because this was sort of key to the inferences that they were making. And even with this good model, these really nice data, this great sampling, at some point you're still making an inference based on your subjective interpretation of what these values mean. And I think we've all struggled with that, right? We've all gotten an estimate of some parameter like migration rate and asked ourselves, well, what does this mean? If it's 0, that means something really definitive to most of us. But usually these estimates are slightly more than 0, and we have to interpret how we're gonna sort of deal with that and think about that and make inferences then from these estimates of parameters. So regardless of whether we're using relatively model-free assumptions or with these more complex statistical phylogeographic models, we're making some subjective or qualitative decision about the inference that we're taking out of the data. And as Knowles and Madison pointed out in their statistical phylogeography paper, anytime we do this, the natural tendency is to make what they termed an overinterpretation. And I sort of take this to mean we work really hard to collect the samples and collect the data and submit the paper and revise the paper and in some cases revise the paper again. So we want it to mean something. We want to tell a really detailed story about the history of our species. And when they say overinterpretation, I think the meaning here is that our tendency is to tell a more detailed story than maybe the data can really support. The other problem with this is a phenomenon called confirmation bias. And this is something that psychologists are really aware of. And so there's a paper by Nickerson. And they talk about this phenomenon that humans have where we interpret new information in light of what we think we know about something. And I think this is something that we're constantly guarding against in my lab. We're guarding against this tendency to assume we know something, make an estimate of some parameter or an inference from it, and then interpret what we think we know. So a good example of this, again, is gene flow. If you're estimating gene flow among what you think are populations of the same species, a certain value of a parameter might be evidence for gene flow. But if you think these things are separate species, then maybe you wouldn't look at this value as being high enough to really merit being indicative of gene flow. And so we have this confirmation bias based on what we think we know. So when we do analysis in phylogeography, our goal is really to make inferences about the evolutionary history. And I think the great contribution that all of us who are doing research in these empirical systems can make to the broader field of genetics is that we're working on things where we don't know the answer, right? I think human population geneticists have a pretty good idea about the basic demographic history of human populations. We don't have the luxury of all of this. We also don't have the ability to do replication of evolutionary history. We don't really have experimental controls. And our discipline is very much one that is historical in nature. And we struggle a lot with how we should treat the data statistically. And I'm going to try to convince you today that we shouldn't rely on statistical tools that were developed in the experimental sciences, but that they're better options to use because we're working on a historical discipline. And the main approach I'm going to take is to argue that if we proceed by trying to choose the best of many different models, this is a compelling framework for making inferences in phylogeography. And the reason for this is that we don't really know what the demographic history is, and in many cases, this is the key piece of information that we really want to get out of the analysis. And so if we can find ways to calculate the probability of a range of demographic histories, given the data that we can collect, and if we can find ways to identify which of these probabilities is the highest, and maybe even assess some level of statistical confidence in that, we've gone a long way towards making the inferences that we want to make. So there's several approaches to this, and I want to just sort of walk you through what I guess are maybe 3 different approaches that are all valid and useful in different situations. One of these is the type of statistical phylogeography that Lacey and Wayne Madison described in their 2002 paper. And this figure itself is from a paper that I did with Lacey while I was her postdoc because I had the figure on my computer still, which was kind of scary. But basically what we did here is we calculated these null distributions, and this was based on the number of deep coalescences for 2 loci. So this was 2007, we were trendsetters, right, with 2 loci. And we compared the estimate of this parameter from the empirical data to the null distributions. What we basically were doing here is a type of hypothesis testing. And so we were calculating the probability of our data given the assumption that the model was true. And when we basically estimated the p-value of our data given this null distribution, we were learning something about how well these different models fit the data. This is great, but it's also based on a long list of assumptions about the aspects of the model. And so in this case, we had theta values that we were assuming. We were making assumptions about our sampling strategy and the timing that was involved in the diversification, et cetera. And so when you reject a model like this, it's sometimes difficult to understand what that means. If you fail to reject a model like this, it tells you that your data are a pretty good fit. But when you have empirical data that are off on the tails here, you know that your model doesn't fit well, but it's hard to identify why it doesn't fit well. There are, of course, other approaches to fitting these complex models, and an extremely useful one is ABC. And I want to talk about these other approaches after mentioning a review paper that I both really like and I'm really frustrated with at the same time. So this is a paper that was published by Nielsen and Bowen in 2009, and they're arguing for making inferences in phylogeography based on these complex statistical models. And at some point, towards the end, there's a paragraph that I always imagine was put in at the request of a reviewer that basically says a clear limitation of any model-based method is that the model might be wrong. In fact, the real complexity of the demography of natural populations is unlikely to be captured by any simple model we could propose. And so you're reading along and you think, well, okay, I sort of know that. It's this next sentence that really does it for me. And they say, in some cases, this might not affect inference as much, but in other cases, it will. And then they change the subject, and that's the end of that. Thanks, guys. Yeah. And so this has really stuck with me in the last 5 years or so, and we've really tried to think about ways around this question. So what can we do if we want to use model-based inferences? We want to use models that are to some extent tailored to the particulars of our system. We've read this paper, so we know it's important to think about model fit. Well, an easy approach, or at least a workable approach, is to actually assess the probability of a number of different models given the data that you can collect. So there are several good examples of this in the literature. Many of these use a Bayesian framework, and they rely on approximate Bayesian computation. And so I think most of us are probably familiar with this well-known paper by Fagundes et al. That tests different models of human demographic history. And so what they do is they have some multi-regional models, some out-of-Africa models that have been informed based on the information that's available in humans. And they calculate first which of the out of Africa or multi-regional models that they can parameterize is the best fit to the data that are observed, and then they compare the best of these models. And essentially what you're doing when you do this type of approach is you're calculating the posterior probability of your data given the model and the priors. And so it's a reversal of the relationship between the data that you collect and the model that you specify. Unlike a frequentist approach to the hypothesis testing where you calculate the probability of your model given the data, and thus the assumption that the data are true, or that the model is true, here you're reversing this relationship. And so this is an extremely useful approach, and it's something that can be adopted to almost any system. The only disadvantage of it is most of us don't work in systems where they have entire departments devoted to collecting non-genetic data that tell us information about the demographic history of the population, right? So if you're working in frogs from South America, for example, we know a little bit less than human geneticists do about formulating these models. And that's not to say it's impossible to do so, but it's an additional challenge. And I think one of the great sort of contributions that we're going to make in the next 10 years of phylogeographic research is to really think about how we can identify models that are reasonable to test in our non-model systems, that have all of this diversity that far exceeds the diversity that we would see in a model system. Another approach is very similar to this, but it has a different statistical framework. And this is the one that we actually favor in my lab. It's an information theoretic approach. And the basic way that we do this is if you have an approach or an analytical method that allows you to calculate the probability of the data given the models in a likelihood framework, and you don't have to rely on the prior, it gives you a little more ability to discriminate among a larger number of models. And so we have an example of this from a few years ago where we did model-based inference in a group of willows, and we had a dozen or so different demographic models that we could evaluate. And so what I'd like to do today is quickly take you through how we've done this in this frog from the Caatinga of northeastern Brazil. This is Pleuroderma allium and Diplolister, and this is work that has been done primarily by Teresa Tomei, who was a postdoc in my lab last year, and who we've continued to work on some of these questions. And so Teresa went out and has done a really amazing job, not only with this species, but with several other species of frogs that are from the same area. And she's now sitting on a great comparative data set that she's working on analyzing. And so I am not going to pretend to be an expert on the Caatinga. There are people in this room who know more than I do, I am sure. But basically, it's part of this dry diagonal in South America that's between the coastal Atlantic rainforest and the Amazon that we heard about in Anna's talk earlier. And so it's this xeric environment that's surrounded by mesic biomes. It's really unpredictable and highly seasonal. And so there's years that are very dry and have severe droughts. There's other years that are wetter. And so these are pictures from a similar perspective in different, wet and dry seasons, just to give you a feel for this. And there's been a lot of research into the dry xeric areas in South America and trying to understand their origin and how they've been affected by climate change and sort of responded to changes in the wet forests, et cetera. And we know that the Caatinga is one of the seasonal dry tropical forests, and it's one of several isolated nuclei at the present. There's been a lot of both fossil work and paleo modeling work on this. And so you can find examples in the literature that would predict that the habitat in the Caatinga was very stable over recent evolutionary time and also very unstable. So there's this debate, depending on how you do the modeling, about the extent to which this habitat would be broken up by wetter mesic elements as a function of climate change. There's pretty good record from fossil pollen and other kinds of fossils for recent wet forest invasions into the Caatinga. And so Teresa's motivation for going out and collecting a bunch of amphibian samples from this region is primarily one where she wanted to understand if these frogs, which were adapted to the xeric environment, had been affected by all of these forest invasions that you can find evidence for in the geological literature. And so I think when she went into this project, and certainly when we were talking about it, when she first emailed back and forth about visiting the lab and coming to work in the lab, the idea was that there would be different barriers in some ways that might promote population genetic structure. And so we're now starting to finally get the first papers from this work out. We have one that's in press in Journal of Biogeography, and I just wanted to give you a background based on microsatellite data and CO1 data of what we could infer from these sort of more traditional markers, and then I'm going to talk about the RAD-seq data that we're working on at present. So the first thing that Teresa did was collect CO1. Right? It's the first thing that a lot of us do when we start a new project. And that's because it's still a really useful marker. And so here's a haplotype network from the mitochondrial data. She collected something like 350 frogs, just an incredible number of them. And the important thing to recognize is that the two species, Allium shown in pink and Diplolister shown up here, are separated by a lot of mutations. And so they're quite distinct at the mitochondrial gene. This is a structure plot based on 12 microsatellites, and the two species form two different groups. And so there's no evidence based on these data that these are anything but the two species they're described as. What's really interesting is in Diplolister, which is the species shown in black that has a widespread distribution, the only population genetic structure we can detect within Diplolister is a structure that is sort of sympatric with the sister species, the alien. And this seems really strange, and we sort of thought about different speciation models, or maybe it was something like a peripatric situation or something that would lead to that pattern. But going into the RAD-seq analysis, these were the things we knew. And what we wanted to do then is to collect RADseq data to test more detailed models of the history between these species and within these species, and also try to learn something about the timing of speciation, population sizes, whether they had expanded after the Pleistocene, and these types of inferences. So this is my really boring slide that I feel obligated to put up because I want to make sure that I don't gloss over a lot of the details that I'm going to gloss over anyway. But we collected RADseq data, and we only sequenced 192 of the 350 or so individuals. But that's still a pretty good sized data set for RADseq. I think the sequencing reads were like 476 million or something. We used a variety of software packages, including PyRad and the Genome Analysis Toolkit and Python scripts that Teresa wrote to edit, align, quality control, SNP call, et cetera, all of these data. And what we end up with in total across all of the samples that were collected is something like 6,200 aligned loci that contain SNPs, representing about 500,000 base pairs of total sequence data. And so when I talk about how we're now in the position where we can close the gap between non-model work in genetics and model systems, clearly we're not there yet, but 5 years ago you wouldn't have been able to collect these data from relatively unknown frogs nearly as easily as we could. We then did a number of analyses trying to get a basic sort of a handle on what we had. And the interesting thing was we didn't identify any population genetic structure within either of these species based on the RADseq data. We ran structure 8 different ways, and it always came up, there was no structure, which in some ways makes things easy. In other ways, Teresa went into this expecting that there were these forest invasions that were going to lead to population structure in the species. And I think she still isn't really convinced that that's the true answer. But we're moving on. So the other thing we were really interested in doing, because we have this case where the sister taxa are partly sympatric, it seemed like there might be some competitive exclusion or some adaptive diversification or something like this. And so we spent a lot of time looking at the SNP markers to identify SNPs under selection. We've used several approaches. I just want to show you a few examples of results from Bayes-scan that are representative of what we've seen. So Bayes-scan is this basically outlier detection model. And if we treat all of our sampling localities as populations, which is sort of it would bias us towards identifying a large number of SNPs, we find a really small percentage of outliers in the grand scheme of things, less than 5%. And this, again, is probably not the best way to do it because we don't have any population genetic structure. And so to treat all of our sampled localities as populations is clearly not what we want to do. If we do something much more conservative, like just look at the 2 species and treat them as 2 different populations and do the analysis, we find a very small number of SNPs. We think probably the best thing to do is to have 2 units in each species based on whether or not they're sympatric or not with their sister taxa. And so if we divide things like this, our plot goes from 217 outlier loci, which is still a small percentage of the total, to, 18? So it doesn't appear that selection is really driving the pattern of genomic diversity that we see in these SNP data, at least. And it also, it doesn't appear that there's lots of population substructure. And so the question then becomes, what is the driving force here? All of these analyses are really complicated by missing data. And the one thing that we have really sort of struggled with in my lab in the last 2 years or so is how we can make good inferences based on SNP data without misleading ourselves due to the missing data. I just want to give you a flavor for this by showing you what hopefully is an easy to understand graph. So the y-axis here is the number of SNPs that we would identify, and the x-axis is the total sequencing reads. And each of these dots is an individual that we've sequenced to some level of coverage. The gray areas here are showing the SNPs that we would call if we didn't worry at all about missing data. So even if we only have 2 individuals at some locus that are sequenced and they have a SNP, we would call that. The black dots are the number of SNPs that we would call if we had a threshold like 70% missing data. And then these ones that are outlined in red are individuals that just didn't work well in the library prep for whatever reason. Most of these samples were older samples that weren't collected by Teresa. She got them from museums or something, and so the DNA quality wasn't as good. Probably the DNA was fragmented, and it didn't work well when we did the library prep. So the question is, what do we do with this? One, sort of common approach is to just say we're gonna have some cutoff, and we'll just analyze these data that are present above some threshold. But I think the problem with doing that is there are many methods that we're wanting to use that rely on some information about rare alleles. And as Scott mentioned in his talk, there's good reason to think that you bias your loci away from things that might have mutations and restriction sites, and so you have like the sort of initial bias. You're also not as likely to detect rare alleles when you allow a certain amount of missing data, et cetera. And so this has become, I think, a really challenging question for us to address as a field. To give you a flavor of this, these are just examples from a very small number, but here's all of the SNPs in this table, and I've put the fields in pink or whatever, red, that where we don't have data for because that individual didn't have a locus sequence that the 10x coverage or whatever we required. We could just go ahead and remove all of the individuals that have missing data, but if we do this, we go from a relatively large number to very few individuals. And of course, as you have more and more loci, this number gets smaller and smaller. So that's not really a good option. We certainly don't want to do something like this where we take every locus out where there's missing data. Because now we're going from our really nice data set with thousands of SNPs to one that has 3. And I think even if it's hard to publish a paper in good journals with just mitochondrial DNA these days, I don't think a paper with 3 SNPs is going to go far either. And so Jordan Sackler, who's one of my PhD students, his picture's up there, has really pioneered an approach that we think is very effective for this. What we do is we downsample our data. And so we go through and pick a number of individuals and loci that we can call in our data that are represented in all individuals. And then we can do replicates, and we can use these downsampled data sets rather than our full data in the analyses that we're making. And so to give you a feel for what happens when we do this, we go from something like 200 individuals and 6,000 unlinked SNPs to maybe 100 individuals and 3,000 unlinked SNPs or something. And so it's less data, but it's still a lot of data. And we go away from this missing data question in a way that hopefully isn't going to bias any of the results that we get. That's the theory, at least. We will see what the reviewers think when Jordan submits this paper. But I'm going to go ahead and use his script anyway, because we felt like we wanted to have some results to show. So what we've done then is we've used this program that Laurent Escoffier's group has been working on called FastSimCol2. And this program uses a combination of simulations and calculations from the site frequency spectra to calculate the probability of our data given a variety of models. And what I like about FastSimCol is it's very flexible. And it computes a composite likelihood, and as long as your SNPs are unlinked, then there's really no reason why you can't compare models using this composite likelihood. And so basically, the other thing I like about this is at its root, the simulations are based on gene tree distributions. And so this goes back to work that Rasmus Nielsen did like in 2000 or something like this, where you can calculate expected distributions of site frequency spectra based on genealogical patterns. And so even though we're not directly using gene trees, this information is still derived in some sense from those expectations. So now that we have a really big data set and a program that's really flexible because we can specify almost any model we want, the question becomes, what models do we specify? And I would argue that there's 5 or so major components that we could put into a model for these frogs. Based on the difference in the range, we might expect that the population sizes between these 2 species differed. Because they're different species, we know that we need to parameterize the divergence between them, this population divergence, lineage divergence. In the CO1 data and the the microsatellite data, there's evidence for population expansion, and so population expansion is something that we might reasonably parameterize. There's also some indication in the microsat data that there's population genetic structure in one of the species. There's not a huge indication of gene flow other than a few admixed individuals, but that's always a possibility as well. And so we could build this model to try to include as many of these parameters as we wanted to. And then we could make estimates of the parameters based on this full model. And when we do this, it works perfectly well. We get a probability, we get some parameter estimates. But we're not working in a model system like the human geneticist. And so the question then is this really the right model to use? The thing about FastSymCol is you can specify any model that you can imagine between these diverging species. And so this is one of literally hundreds, if not thousands, of models that you could draw down and code. And so the challenge then is to understand not what the parameter estimates under some model are, but what is the best model that we should be using to estimate the parameters in the first place. And to try to do this, what we've decided to do is specify a number of models that hopefully cover the range of different types of models that we can imagine. And so in this previous slide where we have these different processes that we can parameterize in the model and that represent real evolutionary processes that we're interested in this system, we'd like to have models that incorporate different combinations of these processes. And that's what I've shown in this slide. Here's 9 additional models, and you'll notice that some of these include expanding populations, some of these include gene flow, some of these include population bottlenecks. You're only limited really at this point by your imagination. And what we can then do is we can calculate the probability of these models given our data, and we can compare this probability. And when we do this, we get a really great result. These numbers don't mean anything to anyone, but 3 models have much better log likelihoods than the remaining models. And so the likelihoods are shown here. These models have differing number of parameters, so we can calculate AIC scores based on the likelihood and the number of parameters. We can then convert these AIC scores into AIC differences and ultimately into this WI. These are model probabilities, and they're directly analogous to posterior probabilities in a Bayesian context. And so essentially what this does is it tells us that, 6 of these models have 0 model probability. So there's no reason to think that they're good models. And 3 of the models have between 0.22 and 0.54. And at first I was really disappointed because I wanted there to be one model that was so clearly the best one. But take a look at what these models are. These are all isolation with migration models. And they're actually probably not really as different in actuality as they might appear to be at first glance. One of them is a model where there's gene flow in both directions that starts when the populations begin to diverge and continues. The second is a model where gene flow is only occurring in one direction. And the third is a model where gene flow starts at some point after divergence and then continues in both directions. When we estimate the time of migration, it's closer to the divergence than the roots. So these models are maybe a little more similar than we would think. But it doesn't really matter. So there's some uncertainty in the models that we've used. We can make parameter estimates and do it in a way that takes that uncertainty into account. And so basically, you can estimate a lot of the same parameters in these different models and then weight these averages by the probability of the model. And so if you estimate population size in model 4, which I think is this purple one that has about 56% of the model probability, whatever your estimate is here, you can weight it by that value. And this gives you a way to sort of get rid of the uncertainty in the model if you want to think about the parameters that you're estimating. And I've done this in this next slide. I just wanted to remind you, we have this full model that includes a bunch of extraneous information. And importantly, it includes populations that are expanding and different, an initial amount of population size and an end amount. And what these extra parameters are doing is they're inhibiting your ability to estimate the parameters that are really important to the system as identified by the results of the model selection. And so if we model average the parameters that are important to estimate, what we find is that we've converted all of these assuming a mutation rate. That's probably wrong, but what are you going to do, right? It's a frog. So it's a reasonable rate based on the literature, and we can convert divergence time into years, and it's about 57 years before present. So a relatively recent divergence time. relatively small ancestral population, and P. allium is a relatively small population into the present. But this other species that has a really big range has a much larger population size. And so this is reflective of the actual difference in genetic diversity that we observe. Rates of gene flow, I've only shown one here, but are much greater going into Diplolistor than to allium. And so this is consistent with the pattern that we're seeing in the model selection. Take a look at what happens when we compare this, which I would argue was a good model to use for this system, to the full model. And just compare the difference in parameter values for some of these parameters. Look at divergence time. It's 2 orders of magnitude different. And so I don't know anything about these frogs, but it's easy enough to sort of, we're talking about the difference between late Pleistocene diversification and pre-Pleistocene, significantly pre-Pleistocene. The population sizes are very different as well. Rates of gene flow, are very different. Now, when we look at these parameters, it's easy to think, oh, well, that's a bad model, so we wouldn't expect there to be good parameter estimates. But what I would argue is that if we're not doing some kind of model selection exercise, how do you know that the parameters that you're estimating aren't closer to that than something that might be better justified? And so we have, I think, a unique challenge working in non-model systems. And we're working in things where we really don't know what the answer is. What I find amusing about FastSimCol is that it's developed to work, the test data are human data. And so when Escoffier, I thought I had a slide, I had it. When Escoffier tested it and describes the test in the paper, they basically write, the ability of FastSimCol to estimate parameters was compared against known parameters values from humans. And so their quality testing was basically, we know the answer, so let's see how close we can get to it. This isn't a luxury that the rest of us have. So what about the other pieces of information that we might use to support this diversification model? There's very little difference in the ecological niche between these species. This isn't surprising because they're sympatric, so they have a lot of niche overlap. So this, combined with our lack of SNPs under apparent selection, suggests that the divergence isn't really adaptive. This must be a case where these things are periodically isolated and maybe either come into secondary contact or limited contact where they exchange genes. This is somewhat supported by the ecological niche modeling that Teresa has done that show the really dynamic nature of this Katenga habitat and how it changes from the current suitable range to the mid-Holocene and the last glacial. So I want to close by sort of stepping away from the frogs for a minute and talking about the data that we can collect. And Scott mentioned this paper that Ryan Garrick's lab and our lab worked on. And to the surprise of absolutely no one, we were able to demonstrate that the number of loci, number of SNPs, however you want to measure it, in phylogeographic data per study is increasing. And increasing fairly dramatically. This is a log scale on the y-axis going from 1992 to 2013. But what's really encouraging is that we've done this in non-model systems, and molecular ecology, I would argue, was publishing sort of representative results from non-model systems as a rule, without sacrificing the number of individuals that we're sampling. And so the strength of phylogeography has always been that we sample lots of individuals in our studies. And this number of individual samples hasn't changed even as we've gone from a single locus to now thousands of SNPs. And so that's, I think, really good. It's encouraging for the future, and it's going to allow us to really ask different kinds of questions in our non-model systems that might be important in model systems. And this is also changing what's important to think about. So as a grad student, I was doing my dissertation right at the time when we were beginning to realize that a single locus wasn't enough data. And I would read papers like this paper that Scott and Peter wrote, or the Brumfield et al. Paper, about how you don't need one gene to estimate parameters, you need 50. Or in the case of this Felsenstein paper, he argued that you would be better off sequencing thousands of individual sites than one site that was thousands of base pairs long, and we all laughed and said, how could we ever do that? And of course, we can easily do it now. By the time I was done being a postdoc, I had carpal tunnel syndrome because we had to collect these data one at a time. And now we can almost get the same quality of data in any system we want to investigate as you can in the most developed model systems. And this is a really encouraging development, and it's amazing that it's happened as fast as it has. I would also argue that we're doing a lot of deep thinking about models in phylogeography, and that non-model people working in empirical systems are really leading the charge in this. And so I've thought about the different ways that people are asking questions that can be categorized as model selection in phylogeography. There's now an expansion in Migrate that allows you to evaluate different migration models for systems. ABC is, of course, really useful for this, and there are now great R packages like the one that Kate Sillery put together, trying to make it easier to build ABC pipelines in non-model systems. There's examples in SNP data, like I've talked about. I'm working on a project with Brian O'Mara called FRAPPLE that's trying to do model selection with gene trees. And so for just a single species, there's lots of interesting ways to do this. You can think about a lot of things with species delimitation as ultimately being model selection as well. And of course, phylogeography has always been interested in where the limits of species are. And there are now a number of methods, including BPP and ABC approaches, and the approach that Adam Lachey is working on. There's a nice new approach that uses both morphological and sequence data. And they're, in one way or another, based on this idea that you can choose the best model between separate or the same species. I would also sort of suggest to you that the hierarchical models that are being pioneered in Mike Hickerson's lab, and among other places, are in some sense model selection, just in a comparative framework. And I'm really excited about new approaches to this. So there's a paper that Xander, is it out yet or it's not out yet? It's out, okay. It was just published using these hierarchical models for testing for simultaneous population expansion. And the great thing about these models is that they're really flexible, and you can apply them in different ways to a variety of questions that we're interested in. Oh, there's the slide I was looking for. So here's the known values for Escoffier in the human systems. And I would argue that unless you're working in a system where you know the answer already, one of the first things we want to do for phylogeographic studies is think about how we can identify models that are good fits to the data. It's more important to understand which parameters we should be estimating, and unless we're really able to objectively identify the model and the parameters that are important to estimate in a system, we shouldn't be worried as much about what quality of the estimates. And so I think in the future, what we can contribute to the broader field of genetics is really good, creative, and innovative ways of identifying models that are a good fit to particular questions in particular systems. So the other thing that we can do is we can think about assessing absolute fit. So now that we've identified this model that's the best model in our system, we are working on doing things like absolute goodness of fit tests. And so this is something I had hoped would be done, but it takes longer to analyze than I had expected. So hopefully by the time the paper is due, we'll have results. So to wrap up here, you know, the big limitation of model-based methods is that the model might be wrong. And what I would argue is that this is the question that we really want to be addressing when we start working in empirical systems. So I'd like to thank a bunch of people who've worked in my lab, either in the present or in the past, but in particular, Teresa Tomei, who did the collection of these data from the frogs, and Jordan Sackler, who helped with the downsampling script. And I'm happy to take any questions.